Welcome to our deep dive about selective and differential media. I want to just start by asking you a question. Have you ever wondered how a doctor's clinic takes a simple throat swab, or maybe a municipal lab takes a murky water sample, and then somehow, within a single day, they know exactly which dangerous bacteria is lurking inside?
Right. Because, I mean, the natural expectation is definitely, a room full of glowing screens.
Yeah, exactly.
Like, they're running these complex algorithms to crack the genetic code of the pathogen. We're just so conditioned to picture modern medicine as sleek and digital.
But that high-tech illusion shatters pretty quickly when you look at how clinical microbiology actually operates day to day. The foundation of sorting and identifying these invisible killers, it doesn't rely on a microchip. It relies on this brilliant, century-old method of using, well, chemically spiked jelly to force bacteria to reveal their true identities.
It really is the ultimate low-tech hygienia solution. I mean, to this day, it's still the absolute fastest and cheapest way to answer the two most critical questions in a diagnostic lab, which are, what is growing in this patient and how do we kill it?
Right. And that brings us to the mission for this conversation. We are going to decode this brilliant system that microbiologists call selective and differential media. We'll get into the incredibly clever lab tricks they use to pull one specific pathogen out of a microscopic crowd. But to really grasp how this invisible sorting works, we have to start in a German kitchen in the 1880s.
Oh, yes. Because before you can sort bacteria, you need a physical stage to put them on, right? You
Right.
can't easily sort a chaotic liquid soup of germs.
Right, it's just a mess.
Exactly. Trying to find a specific bacterium in a liquid broth is basically like trying to study a single fish in the entire ocean. To properly study a pathogen, you need what scientists call a pure culture. That's a single species isolated from everything else.
And in the early 1880s, Robert Koch's famous microbiology lab was desperately trying to do this, right? By using nutrient gelatin.
Yeah, the concept was simple enough. You pour a liquid nutrient broth laced with gelatin into a dish, you let it set into a firm surface, and then you just swipe your bacterial sample across the top. The individual bacteria land, they get stuck in place, and they multiply into visible, isolated mounds called colonies.
I mean, I see the logic, but knowing a bit about gelatin, there had to be a massive physical limitation when they tried to incubate these plates at human body
Oh,
temperature.
it was a total disaster.
Yeah.
Yeah, because human pathogens generally need to be grown at human body temperature to thrive, right?
Right.
Which is 37 degrees Celsius. And gelatin melts well below that. So the moment they put these dishes into the incubator, that nice firm surface just turned into a warm, useless puddle,
Oh, wow. So they just had soup again.
basically. And adding insult to injury, many bacteria produce proteases, which are enzymes that break down proteins. Since gelatin is a protein, the bacteria were actively digesting the very floor they were standing on.
They were literally eating their own stage.
Yeah.
So the researchers are stuck with this soupy, contaminated mess. And the solution didn't come from a chemist, did it? It came from Fanny Hess.
Right. The wife of one of Koch's assistants.
Yeah, and she changed the entire trajectory of medical science with a cooking trick.
She really did. To keep her puddings and her fruit jellies firm during the hot summer weather, she didn't use gelatin, she used agar. It's a complex carbohydrate derived from red seaweed that had been used in Asian cuisines for, centuries.
I just love that the foundation of modern microbiology, the actual tool that allows us to fight infectious diseases, was essentially saved by a 19th century dessert hack.
It's amazing. And the chemistry of agar is practically miraculous for this specific job. It exhibits this property called hysteresis.
History.
Yeah, meaning its melting and solidifying temperatures are vastly different. It won't melt until you heat it to about 85 degrees Celsius. But once it's melted, it actually stays liquid all the way down to about 40 degrees Celsius before it sets.
Wait, why is that temperature gap so important for the scientists mixing this stuff up?
Because you have to mix in highly sensitive ingredients, things like specialized sugars, antibiotics, or even whole red blood cells. If the agar had to be boiling hot to stay liquid, you would just cook and destroy those delicate additives the second you stirred them in.
Oh, right.
Yeah, with agar, you can let it cool to like a nice warm bath temperature, stir in your fragile ingredients, pour it, and then let it set. And furthermore, because agar is a complex plant polysaccharide, almost no human pathogens possess the enzymes to digest it.
So it's biologically invisible to them.
Exactly. Add in the shallow covered glass dish invented by Julius Richard Petrie in 1887, and the physical hardware of microbiology was finally finalized.
Okay, so let's unpack the chemistry now. Scientists had this solid, reliable agar canvas, but they still had to figure out how to sort the microscopic mess growing on it. I mean, a throat swab or a murky water sample contains hundreds of different types of microbes.
Thousands easily.
Right. Finding the one dangerous needle in a haystack of harmless bacteria requires some intense manipulation.
And manipulating that agar is where we get to the core of our deep dive: selective media and differential media. These are the two primary strategies used to force the bacteria to reveal themselves.
Okay, I was actually trying to visualize this while going through the research, and I came up with an analogy. Tell me if this accurately captures the science.
Sure, let's hear it.
To me, selective media is like a highly strict bouncer at a nightclub. The bouncer's entire job is to check IDs at the door and turn certain groups away, while letting a specific group inside. It asks the fundamental question, who gets to grow here at all? On the flip side, differential media is like handing everyone who makes it inside the club a different colored glow stick based on their favorite drink. Lets everyone in, but it asks, of the crowd that successfully grew, which one is which?
That analogy holds up perfectly on a biochemical level. It really does. Let's break down how your bouncer and your glow sticks actually operate in the lab.
Okay.
So selection the bouncer functions by weaponizing environmental stressors. The agar is spiked with ingredients that exploit the structural vulnerabilities of certain bacteria. Take mannitol salt agar as an example.
Okay.
The lab pumps the agar full of a 7.5 to 10% salt concentration. If a typical bacterium lands on that plate, the extreme salt environment causes the water inside the bacterial cell to just rush out through osmosis.
Because it's trying to balance the salt.
Exactly. So the cell essentially shrivels up and dies.
But the bacteria we're trying to find must have some kind of armor against that, right?
They do. Staphylococcus species, for example, naturally live on our salty, sweaty human skin. They've evolved robust cell walls and internal pumps to manage extreme salinity. So while everything else shrivels, staph thrives.
That is so smart.
Alternatively, the bouncer might use specific targeted antibiotics. Like if you're hunting for a drug-resistant fungus, you might lace the jelly with penicillin to wipe out all the competing bacteria, leaving a clear field for the fungus to grow.
So the bouncer uses salt, antibiotics, or extreme pH levels to literally poison the microbes we don't care about. Then we have the glow stick's differentiation.
Right. Differentiation relies entirely on exploiting metabolic differences. You provide a specific food source in the agar, usually a type of sugar, and you pair it with a visual indicator, often a dye that responds to pH changes.
Okay.
If a particular bacterium has the genetic instructions to ferment that specific sugar, it'll consume it and produce acid as a waste product.
And that acid drops the pH of the jelly locally, right? Like right around that specific colony.
Yes, the acid alters the chemical structure of the dye embedded in the agar, causing it to change color. So if 10 different types of bacteria are growing on the plate, the one species capable of fermenting that specific sugar will suddenly light up bright yellow or pink or red.
Wow. The brilliance of this really comes into focus when you look at why it was invented. This wasn't just, scientists tinkering for the fun of it. The development of these chemical sorting plates was driven by sheer panic during a public health crisis.
Oh, absolutely. The real catalyst was the struggle against waterborne epidemics around the year 1900. In Britain, officials were trying to contain devastating outbreaks of enteric diseases like typhoid. To do that, they had to test drinking water, sewage, and feces.
I can only imagine the sheer volume of microbial noise in a sewage sample. Finding the bacteria that causes typhoid salmonella right in untreated sewage must have been an absolute nightmare.
It was equivalent to finding a specific grain of sand on a beach. Every sample was teeming with millions of harmless environmental bacteria and normal gut flora. In response to this crisis, a bacteriologist named Alfred MacConkey developed what we now call MacConkey agar, publishing his formula in 1905.
Right.
It was a total masterstroke because it combined both your bouncer and your glow stick into a single, elegant plate.
Let's look at how he built this dual system. What was the bouncer in his recipe?
So, MacConkey used bile salts and crystal violet dye. He reasoned that the dangerous pathogens he was hunting for, like salmonella, naturally live and infect the human gut. The human gut is filled with bile, which acts like a harsh detergent, basically tearing apart the lipid membranes of many bacteria.
Okay, so gut pathogens are already adapted to tolerate bile.
Exactly. But gram-positive bacteria, a massive category of microbes that lack a protective outer membrane, they are completely shredded by it. So the bile salts suppress the non-gut bacteria, clearing out the noise.
Using the host's own digestive chemistry against the competing bacteria is incredibly smart. So the bile salts clear the way for the gut bacteria to grow. How did he hand out the glow sticks to figure out which gut bacteria was which?
For the glow stick, MacConkey added a sugar called lactose and a pH indicator called neutral red.
Lactose, like in milk.
Right. Lactose fermenting organisms, such as harmless strains of E. coli, devour the sugar, they pump out acid, and they turn the neutral red dye into a vibrant pink or magenta.
Oh, wow.
However, dangerous non-fermenters like Salmonella or Shigella, they lack the enzymes to eat lactose. They grow happily on the plate because they survived the bile bouncer, but they stay completely pale and colorless.
This actually connects to one of the most famous medical mysteries of that era. I mean, we've all heard the story of Typhoid Mary, Mary Mallon.
The cook.
Yeah, she was a cook in New York in the early 1900s who left a trail of typhoid outbreaks at the wealthy households she worked for. But she was an asymptomatic carrier. She felt completely fine. If Mary wasn't showing any symptoms, how on earth did investigators prove she was the source of the outbreaks without these chemical plates?
They couldn't have. They really couldn't. In 1907, a sanitary engineer named George Soper and Dr. Sarah Josephine Baker tracked her down based on employment records. But you can't just quarantine a healthy-looking citizen on a hunch. They had to prove she was shedding the bacteria.
So they tested her.
They secured her stool samples and utilized early selective culturing methods laced with bile. The bile suppressed her normal gut flora, and the media allowed the pale, colorless colonies of Salmonella typhi to grow and be visually confirmed.
They literally used the plates to make the invisible pathogen visible, proving the concept of asymptomatic transmission for the first time in public health history. That's wild. And the logic behind MacConkey's plate isn't just historical trivia. Pathogens evolve, our food systems change, but laboratories are still modifying this exact same technique for modern threats. I'm thinking specifically about the E. coli O157 and 7 hamburger outbreaks that terrified the public starting in 1982.
The 1982 outbreaks are a prime example of how adaptable this whole system is. E. coli O157H7 is a highly dangerous strain that produces a potent toxin, leading to severe kidney damage. But here was the problem for microbiologists on a standard MacConkey plate. This deadly strain ferments lactose and turns bright pink, looking completely identical to the harmless E. coli that already lives in everyone's gut.
So the glow stick was giving the same color to the harmless crowd and the killer.
Exactly.
So the scientists had to find a different glow stick.
They searched for a metabolic weakness, and they discovered that while normal E. coli can ferment a different sugar called sorbitol, the deadly O157 strain cannot. It is a very poor sorbitol fermenter, so the lab simply swapped the sugar in the recipe.
They took out the lactose and swapped in sorbitol, creating sorbitol MacConkey agar.
Yep. And by swapping that single ingredient, the visual outcome reversed. On this new plate, the harmless E. coli ferments the sorbitol and turns pink. The deadly O157 strain fails to ferment it, and its colonies stay pale and colorless.
That's genius.
During a massive foodborne outbreak where officials need to pull contaminated meat off grocery store shelves within hours to save lives, having a cheap visual screen that instantly highlights the killer is invaluable.
It extends far beyond food poisoning, too. If you walk into an urgent care today with a severely swollen, painful throat, the clinic takes a swab. The test they're running relies on a visual classification system published by J.H. Brown in 1919 using blood agar.
Blood agar is a really fascinating variation because it doesn't use a bouncer at all. It's a non-selective medium that lets almost any microbe grow. But its differential power, its glow stick, is unmatched. The jelly is literally infused with mammalian blood, typically 5% sheep's blood, making the plate opaque and bright red.
And the glow stick here isn't a chemical dye turning pink or yellow. The visual indicator is the actual physical destruction of the red blood cells in the agar.
Right. J.H. Brown identified 3 distinct patterns of destruction. Some bacteria grow on the surface and leave the blood completely alone, a pattern called gamma hemolysis. Others secrete chemicals that partially degrade the hemoglobin, leaving a bruised, murky, greenish zone around the colony, known as alpha hemolysis.
Okay, so that's gamma and alpha.
But the truly aggressive pathogens produce powerful toxins that completely rupture and shatter the red blood cells.
That complete destruction is beta hemolysis. And having looked at photos of this in our sources, it is striking. The bacteria destroy the blood so thoroughly that it leaves a perfectly clear, transparent halo around the colony. You can literally hold the red plate up to the light and see straight through it.
When a clinical microbiologist streaks your throat swab onto a blood agar plate, they incubate it overnight. If they pull it out the next morning and see those perfectly clear, see-through halos, they immediately know they're dealing with a beta hemolytic organism, most likely Streptococcus pyogenes, the culprit behind strep throat. Over a century after it was documented, holding that plate up to the light is still a primary diagnostic step.
I want to pause here because my brain naturally wants to categorize everything perfectly. We've been talking about the bouncer and the glow stick as two entirely separate tools, selective media in one bucket, differential media in the other. But according to the sources, there's a real debate about how this is taught versus how it actually functions in a busy clinical lab. The line is much blurrier than the textbooks imply.
Oh, the textbook distinction collapses pretty quickly in practice. The vast majority of the famous, everyday plates used in a lab are actually both selective and differential simultaneously.
Like MacConkey.
Right, MacConkey agar, which we discussed, has the bile salts acting as the bouncer and the lactose acting as the glow stick. Eosin-methylene blue, or EMB agar, works the exact same way. Because technicians use these combination plates constantly, many introductory courses blend them into a single category.
But pure versions do exist, don't they?
They do, and understanding the pure versions helps clarify the underlying mechanisms. Blood agar is purely differential. It lacks a bouncer entirely. On the other end of the spectrum, you have Thayer-Martin agar, which is purely selective.
Right, Thayer-Martin is the specific plate used to isolate the bacteria that causes gonorrhea, and it is incredibly aggressive. It's essentially a rich chocolate agar pumped full of heavy-duty antibiotics. It kills off the normal flora of the throat or the urogenital tract, allowing only the gonorrhea bacteria to survive. But once it grows, there are no colored dyes. It doesn't differentiate anything. It just acts as the ultimate bouncer.
Exactly. The pedagogical debate centers on whether students should memorize the specific combo plates they'll use at their lab bench, or if they should learn the fundamental mechanisms of selection and differentiation as separate axes.
Understanding the mechanism is crucial because it clears up some massive misconceptions. For example, before reading this material, I assumed a selective plate acted like bleach. I thought it completely sterilized the sample of anything we didn't want.
That's a very common and honestly potentially dangerous misconception.
Yeah.
Selective plates do not sterilize a sample. They merely inhibit the growth of non-target organisms.
Wait till they're still alive.
Sometimes. If a contaminant is exceptionally hardy, or if a technician leaves the plate in the incubator for 48 hours instead of 24, a suppressed bacteria might overcome the stress and form a tiny, struggling colony. The microbiologist still has to apply critical thinking and analyze the size, shape, and context of every colony.
Wow. And there are misconceptions about the colored glow sticks, too. I always assumed the bacteria themselves were turning pink or green, like they were producing a colored pigment. Yeah. But on a plate like EMB agar, where E. coli famously grows with this intense metallic green sheen that looks like a shiny beetle shell, the bacteria isn't actually making green paint.
Not at all. The color is a chemical illusion. It's a dye and pH artifact. The E. coli ferments the sugars in the EMB agar so rapidly and aggressively that it creates a massive localized spike in acid production.
Oh.
That sudden drop in pH causes the specific chemical dyes dissolved in the agar to physically precipitate. They crystallize out of the liquid solution and coat the surface of the colony, reflecting light to create that metallic green sheen.
Which perfectly transitions to the modern frontier of differentiation, a technology that feels like absolute magic, chromogenic media.
Chromogenic media revolutionized the field when it was commercialized in the 1990s by a scientist named Elaine Rambach. It takes the concept of the glow stick and elevates it to a level of incredible precision. Instead of relying on a generalized pH shift, which can sometimes blur or bleed into neighboring colonies, chromogenic plates utilize synthetic designer molecules.
Okay, I have to admit some confusion here because the chemistry sounds incredibly complex. The sources say these plates use synthetic substrates tied to specific enzymes. How does that actually result in a specific color forming on the plate?
Think of the synthetic substrate as a colorless molecule that has a brightly colored dye tightly chained to it. As long as they're chained together, the dye remains completely invisible. Now, different species of bacteria produce distinct, unique enzymes. These enzymes act like highly specific chemical scissors.
Oh, so the bacteria secretes its specific scissors into the agar?
Yes. If a specific bacterium's scissors match the chemical chain of that synthetic substrate, it cuts the chain. The dye is suddenly freed, becomes insoluble, and it precipitates directly inside the bacterial colony, painting it a brilliant, permanent color.
It's a built-in biochemical test happening at the microscopic level.
The clinical application is staggering. Take a routine urine sample suspected of a urinary tract infection. A lab can spread that sample on a single chromogenic plate. If E. coli is present, its unique enzymes cut one specific substrate, painting its colonies bright pink.
Wow.
If Enterococcus is present, its enzymes cut a different substrate, painting those colonies teal or blue-green. If Proteus is present, it lacks the scissors for either, so it stays its natural beige color. You get immediate, unambiguous visual identification of multiple pathogens on one piece of jelly.
These plates are brilliant, they're relatively inexpensive, and they're incredibly fast. But whenever a system seems this perfect, there's usually a blind spot.
Right.
And looking at the history of these agar plates, there is a massive, humbling limitation that microbiologists ran into.
The limitation is profound. It's known in the field as the Great Plate Count Anomaly.
The Great Plate Count Anomaly, named by researchers James Staley and Ellen Konopka in 1985. Break this down because it completely recontextualized everything we just talked about.
Imagine taking a small sample of rich, natural soil or a drop of seawater. If you place that sample under a powerful microscope, you can physically count the bacterial cells. You might visually identify 1 million individual microbes in that tiny sample.
Yeah, 1 million microbes.
But if you take that exact same sample and spread it across the most nutrient-rich, carefully designed agar plates we have in our laboratories, maybe only a few thousand colonies will actually grow.
A few thousand out of 1 million. That is less than 1%.
Less than 1%. The vast, overwhelming majority of bacteria on this planet simply refuse to grow on any artificial medium we have ever devised.
Which completely breaks the entire system we've discussed. I mean, you cannot use a chemical bouncer to select or a glow stick to differentiate an organism that refuses to walk onto the stage in the first place.
The entire system is biased toward the tiny fraction of microbes willing to grow in captivity. We spent over a century perfecting these ingenious chemical tricks, only to realize that 99% of the microbial world requires trace minerals, complex chemical signaling from neighboring species, or specific environmental pressures that Fanny Hess's kitchen jelly simply cannot replicate.
But microbiologists aren't known for giving up. How did they fight back against this limitation?
Well, this anomaly drove a massive surge in environmental DNA sequencing, identifying the presence of bacteria by finding their genetic material in the dirt, even if we couldn't grow them. But it also inspired brilliant physical workarounds. A prime example is the iChip, developed by Slava Epstein and his colleagues around 2002.
The iChip is a fascinating piece of engineering. Instead of trying to blindly guess what nutrients these finicky bacteria want to eat in a sterile lab, they essentially built a microscopic hotel, locked the bacteria inside their individual rooms, and buried the entire hotel back out in the dirt. But wait, if they bury it in the soil, how do they keep the bacteria from just swimming away while still letting the dirt's nutrients reach them?
The secret lies in semi-permeable membranes. The iChip consists of hundreds of tiny microscopic wells. They dilute a soil sample until only one single bacterial cell lands in each well. Then they seal the top and bottom of the chip with a specialized membrane.
And the membrane keeps them tracked.
Right. The pores in this membrane are too small for the trapped bacterium to escape, but they're large enough for dissolved nutrients, signaling chemicals, and moisture from the surrounding soil to flow freely in and out.
It's a diffusion chamber. The bacteria are tricked into thinking they're still living naturally in the dirt, so they happily multiply into a colony, but they're safely contained for the scientists to harvest later.
And harvesting those unculturable microbes has yielded incredible results. In 2015, using the iChip to grow previously inaccessible soil bacteria, researchers discovered a brand new organism called Eleftheria terrae. From that organism, they isolated Teixobactin, a completely novel antibiotic class that we desperately needed in the fight against drug-resistant infections.
It proves how much we were missing by relying solely on the agar plates.
It serves as a stark reminder that while agar plates are incredibly useful, they're ultimately just screening tools. They are not always the final answer. Even in a modern hospital, if a technician sees a faint pink smudge on a MacConkey plate, it might be a slow-growing pathogen, or it might be a harmless environmental contaminant. Today, those visual screens are almost always backed up by modern confirmatory technologies like MALDI-TOF mass spectrometry, which blasts the colony with a laser to read its unique protein signature, or PCR, which definitively detects its DNA.
Bringing this deep dive to a close, it's just remarkable to realize that despite the advent of lasers, mass spectrometry, and DNA sequencing, these clever century-old agar plates remain the indispensable front line of microbiology. Whether a clinic is diagnosing a severe UTI, a hospital is screening for MRSA, or the government is tracking an E. coli outbreak from a local hamburger chain, the first step is almost always putting the sample on a plate.
The agar remains essential because labs often need a living, breathing isolate of the pathogen. You cannot expose a computer-generated DNA sequence to penicillin to see if it dies. You have to physically grow the living organism to test its antibiotic resistance. For that, we still rely on the physical stage Fanny Hess provided.
It's a brilliant legacy. But I want to leave you, our listener, with a final, provocative thought built on that great plate count anomaly. If our very best, most highly engineered chemical bouncers and diagnostic glow sticks only work on less than 1% of the bacterial world, what kinds of radical life-saving antibiotics or completely alien, terrifying pathogens are sitting in the dirt directly outside your window right now? They're completely invisible to us, holding secrets we desperately need, simply because they refuse to eat a 19th century dessert jelly.